Okay, hello everyone. So you might know now where my accent comes from. So I'm originally from Brazil. So I'm very happy to be here and to represent GenScript. You might know GenScript because of our services, but GenScript also has a lot of new instruments and instruments to automate your plasmid purification and protein purification. And that's what I'm going to talk about today. So this slide is just to introduce GenScript to you guys. GenScript has been actually was first founded in 2002 in New Jersey, Piscataway, New Jersey, and we have completed a lot of milestones since then, as you can see in this slide. So let's talk. I want to present to you three new instruments, relatively new instruments, two for automating your plasmid purifications. The first one was the first launched instrument was for maxi preps. We called AmMag Quatro. And the second one is for your mini preps. We called AmMag Quatro Mini. Or 1100, and another one that we just launched, and actually we have this instrument in our booth. This is for protein purification. What unite all these instruments is that all of these instruments are magnet beads based. And I'm going to talk a little bit more about, I'm going to start with the plasmid purification. So a little bit about the history of plasmid is very interesting that although the plasmid was discovered in 1952, it took a while until the first protocols to purify plasmid DNA was finally established. And it would take like 3 days to purify a plasmid on that date. So talking a little bit more about plasmid purification itself, if you can divide the two steps, the two main steps, you first have to lysate your bacterial cells, and the method you most use is the alkaline lysis. So this is the method also our system uses. After you have your lysate, you can do the plasmid purification itself the most common one is ethanol isopropanol precipitation. If you wanted to reach a higher purity, you can do that as well after you automatize your plasmid purification. You can use cesium chloride and a lot of other methodologies. And the most used one is anion exchange columns. Like many of the kits out there, they use this methodology. However, our instrument uses magnetic silica beads, and I'm going to show you later in the next slides how it works. So here is the traditional way to purify plasmid DNA. So you have to go through several steps, including resuspension of your pellets, lysis, neutralization, centrifugation, binding to the column, wash the column, and the column can clog. And if you are handling like several samples, this can delay a lot your work. So what we thought about what is a good solution to skip the purification, the centrifugation steps. So we first thought that we know how to make beads. We know how to make magnet beads very well to coat the beads. And a very interesting characteristic of the beads is the ability to achieve solid phase reversible immobilization, the SPI. And this feature of the beads would skip the centrifugation steps. So why not to implement magnetic beads-based purification in our instrument? So that's what we did, and we are going to show you the two instruments that use this technology. So this instrument, the first one here is the first one launched in 2022, the end of 2022, is the maxi prep instrument. It's a very popular instrument. And I think I see many people here that already have this instrument in your labs. The next instrument was the mini prep instrument. Is all belongs to the Quatro family that we call, or Quatro series, is the mini prep instrument, mini or 1100. So I'm going to first talk about the Quatro Maxi in my next slides. But before I talk about this, I wanted to introduce a quick video. So Quatro maxi is for automated large-scale high-throughput plasmid purification. So this instrument, if you have one controller, you can connect it to 4 modules. In each module, can run up to 6 maxi preps. So if you have all 4 units, you can run up to 24 samples. So, and it's a complete solution because we have all pre-filled consumables. So you don't need to worry about any of the reagents because we provided the reagents. The only reagents that we do not provide is the ethanol, but everybody has ethanol in the lab. So in all the programs, all the protocols are also available. And my team actually optimized the protocols for you so you can get higher yield, higher purity and yield of your plasmid DNAs. So the next slide is going to just tell you a little bit more about the instrument features. So as I told you, it's for high throughput up to 24 samples. The coefficient variance is 3%, so 97% accuracy. Is very flexible in terms of culture volume. And I'm going to tell you about the data you have for different culture volumes and media types. So basically, the instrument works in terms of pellet weight. I would say from 0.6 to 1.5 grams of bacterial pellet would be the optimum to get a high quality of plasmid and high yield. The runtime is 2 hours into 30 minutes. The longest protocol is 2 hours and 30 minutes. And you're going to get a transfection grade plasmid DNA with the endotoxin levels below 0.1 EU per microgram and a supercoil ratio above 80 to 90%. And as I said before, all inclusive consumable kits in a very easy to use interface. So here you have everything in this rectangle here is everything that is fully automated in the instrument. So the only parts that are not automated is the centrifugation of your pellets and the final centrifugation step. So let's go through the steps. So the first step the instrument is going to do is to resuspend, lysis, and neutralize your sample. After this first step, the pre-clear beads are going to be incubated with your sample. So the pre-clear beads are the beads that bind to all cellular debris proteins. And then after the incubation of the pre-clear beads, the magnet is going to come towards to the tube. The magnets are going to is going to be attracted to the magnet, and the clarified lysate is then transferred to the next tube. The next tube, we are going to the liquid handler is going to transfer the binding beads. So these beads are the beads that bind specifically to your plasmid DNA, and then after the incubation steps, you're going to follow by wash steps in the final elution. So in the final elution, if you set it to elute, for example, for 1 ml of your elution buffer, 1 ml is going to be transferred to that tube, incubated, and then the magnet comes again, and then the eluate is going to be transferred to the final collection tube. At this point, is the end of your purification. So the instrument is going to beep. You're going to open the door, take your purified plasmid DNA, centrifuge for 5 minutes, and you have your plasmid DNA ready to use. So this is just some examples about the interface. It's very friendly, very easy to use. Here, the last screen is very nice because it shows which step you are in the process and how long it will take to finalize. So I'm going to show you some application data. So the first application data is very old data. I was just starting collecting data from this instrument. So we compared the AmMag Quatro used PCDNA 3.1, and you compared the AmMag Quatro automated platform with the manual kit. Here, in this case, was the QIAGEN kit. So as you can see, we got a little bit lower concentration comparing to the QIAGEN kit. This was with one of the first not optimized protocols. And then the OD, the 2.6, the ratios 2.6/230 and 2.6/280 was good for both cases. And the levels all below 0.1 U per microgram, so per coil, all above 90%, as you can see in the agarose gel here. So after that, we decided to do a comparison about LB and TB media. So LB media is not very rich. So TB is almost twice richer than LB. And as you can see, with the same culture volume, we got more pellet weight for the TB case. And we using the same protocol and eluting with the same elution volume, of course, we got more concentrated plasmid DNA in the TB condition. So, and all the parameters was as expected. So another study here, as you can see, we use a very rich media that is called Plasmid Plus from Thomson Instruments. And this media actually, with only 30 ml of culture volume, actually we have cases that only with 20 mLs of the culture media, you can get almost 1.5 grams of pellet. And we got the highest yield and highest concentration for comparing to all the other medias we have ever used. So we have another application data here that is comparing during the optimization of the protocols. We have developed a protocol that you could actually retrieve double of the yield. So this is to, oh, I went back, sorry. So this is comparing these two protocols, and as you can see, we could almost double the yield with the optimized protocol that you have now for the instrument. So finally, this study actually was done with a big pharma that is using the Quatro instrument. So when they're validating the instrument, they decided to analyze 2 plasmids. And the way they did that, they sent half of the samples to us, and half of the samples they handled through their manual process. So then, as you can see the data here, with the Quatro, we got a little bit less concentrated plasmid DNA with the first protocol we tried. With the QIAGEN kit, we got a little bit more concentrated. But this is something that you can change in the system. You can elute to, the elution volume is flexible. So you can elute with less elution buffer if you want more concentrated plasmid DNAs. And all the other parameters, the quality parameters was very similar. So then we sent the plasmids, the purified plasmid through Quatro back to them, and they did a side-by-side comparison. So as you can see here in the gel, they did a diagnostic digestion using double digestion and single digestions. And as you can see for the 3 plasmid, comparing to the manual and Quatro purified plasmid, they have the exactly same profile in the gel. So there was no observable RNA contents or genomic DNA contents for none of the preps, either Quatro or manual. So they decided to use these 3 plasmids, purified through these 2 methods and analyze it through transient transfections in show cells. And as you can see here, there is no significant difference in terms of the titer of the protein they use it. They also analyze these. They have automated platforms for 2 transient transfections. They use these 2 plasmids to analyze the titer of the protein after transient transfections. And as you can see here, there is no significant difference for any of the plasmids purified through a manual or Quatro. So with that, I'm going to just go quickly through our new instrument, that is the mini prep instrument. The technology is exactly the same. The steps are exactly the same. What changed is the layout of the instrument. Now the instrument, actually I can quick show you the video for the mini prep instrument. So the beauty of the instrument is that it can be connected to the same controller. So you have one controller that you can connect two minis, two maxis, one mini, three maxis, whatever you want to. So this instrument, the mini instrument, is for a 48 well plate. And you can see the robot arm. You have the mixing functions inside of the instrument. You have the heat plate, so you can control the temperature. The beads that are on the back of the instrument, the reservoir, mix as well, so the beads are not going to settle down over time. And the plate where you're going to have your purified plasmid DNA is going to be a 96 well plate, and you are going to have half of the plate filled with purified plasmid DNA. So talking a little bit more of the features of this instrument, we have here is the 48 well plate. So to start, you have to grow the cultures inside of the 40 well plate. And then you don't need to pour out the media. The instrument is going to handle everything automated from there. So you start with your plate there. You have to fill these containers here, these 4-chamber containers with the buffers that we provide as well, and then put the tips over there in the other vessels, and the bead, and put the beads in the right containers, and then you basically start your purification. And for this, it's going to take 1 hour and 30 minutes to finish your mini preps. And you can connect 4 of these modules in one controller as well. So here's just the workflow. So you start with your pellets here. So you have your overnight cultures inside of the 48 well plates. You centrifuge your cultures, and then you load into the instrument, and you start your process. So after, you're going to have your purified plasmid DNA dispensed in half of the 96 well plate format. This is an example of the protocols that you have several. Is exactly the same as the maxi prep instrument. Each protocol, there are several parameters that you can change. So that's what you can optimize to get higher yield. Depending on the pellet weight, you need to increase the lysis buffer volume, the resuspension volume, etc. Everything is, you can change here, even the magnet beads volume that you are going to use there, and also the dilution volume. So this is a very nice instrument, just launched about 6 months ago. And the beauty of this instrument is actually now we are also already capable of doing protein purification. So mini scale protein purification, you can use the same instrument. We also have a collaboration with Baker Lab that is ongoing collaboration, and they already tested for the protein application. And it's very nice because the instrument can also start with the lysis and go all the purification steps. Of course, with the beads that you are going to choose. If you are using nickel beads, you can use protein A beads or whatever beads you want for your protein purification. And by the end of the process, you also, of course, get your purified proteins. So these are very nice for high-throughput screening of like in mini scale for protein purification. So here is the workflow. I'm not going to go through this because it's essentially the same as the maxi prep instrument that I already talked about. This is the example of the protocols that all the parameters that you can change during the run. And then I'm going to go through some application data really quick. So these first application data, we analyzed the different plasmids in terms of the high copy, low copy, and we analyzed in terms of the yield, how much plasmid you can get with these different plasmids. And as you can see here, we got about 40 micrograms of a high COP plasmid and about 5 to 8 micrograms of a low COP plasmid, and all the purity analysis was in the normal range. So here is another application data that we compared actually 3 different medias growing plates at this for this experiment, and with different with the same protocol. Actually, the protocols are based on the pellet weight, so the culture volume matters in this case. And we analyze, so in terms of the culture volume here, increasing the culture volume, how much of plasmid DNA we can get with this different media. So we can see here that TB gives almost 60 micrograms for a high COP plasmid using about 2 mLs of culture media. Just showing the same thing, the comparison, all the ratios are normal for all 3 medias analyzed. And you also analyzed the endotoxin levels for LB and TB cultures. So if endotoxin level is something that matters to you, we do recommend using LB instead of TB media, so we can get a lot lower endotoxin levels with the LB media. Here we also analyze in terms of the diagnostic digest from the mini preps we ran through the mini instrument, and the supercoil ratio. So here you can see all passed our criteria of being over 80%, and a very negligible genomic DNA by agarose gel analysis. Here is just to show the uniformity of all the samples throughout the 48 well plate in the endotoxin levels as well for all samples. Here is performed a comparison using 3 different plasmids. Here the PTT5, PET28, and back. So comparing with 2 different competitors, so we can see in terms of yield, our instrument gave us very comparable, sometimes even higher yield than the other 2 competitors. We also analyze intransfections using total transfections and EGFP. The protein was expressing GFP, so we did flow cytometry to analyze that, and comparing to the other 2 competitors, exactly the same positive cells percentage there. So, and finally, we just launched this instrument, so I don't have much data to show. And this is the AmMag Quatro 1300. This is for, actually, we have the Assay Plus, so this is the new version of the Assay Plus. Many people have the Assay Plus, and we're also offering an upgrade of your Assay Plus system to this new system. So the AmMag Quatro 1300 we also call ProAb. I don't have a video for that, but we have the instrument in the booth 404. Let me go quickly through this. So this is now it's a fully automated instrument for protein purification. So the basic features of the instrument is for up to 12 samples that you can purify, and up to 8 mLs of settle beads. Any beads that you wanted to use would be fine. A minimum magnetic beads volume of 100 microliters, and a minimum elution volume of 3 mLs. Actually, you can go as low as 1 ml in this case. And you can customize your protocols as well. And here is just the front view of the instrument. You have the controller now attached to the instrument. I think I have other slides showing how it works. So here is, so you have all these racks that they're going to rotate clockwise. This is your sample rack. So you're going to put your beads into the tubes and incubated with your sample. So the instrument has the capability to incubate your sample as well. So you can incubate your sample for up to 3 hours with the current software. So here on the back of the instrument, you have the dispenser. So the dispenser is, of course, connected to up to 6 buffers. There are bottles outside of the instrument with the inlets. So the buffers are dispensed in this station here before the beads are transferred to, so the carousel moves and then the beads are transferred to the next buffer here. So here's just showing the workflow of purification. So you have the incubation with our beads, and also have the magnet rod that's going to be inserted inside of the tube and then can move up and down. So the sample is properly mixed. Then you can set up your wash steps with different buffers. So the buffer is going to be dispensed in the racks containing the 50 or 100 ml tubes. And then you can set up your elution steps, and you can also recycle back or regenerate your beads after the run. So here's a little bit more of the features of the instrument. So we have the dispensing workstation here, as you can see there in A. And then you have the magnet rod. So these 12 magnet rods, and also they have the magnet sleeve. So the rod is inserted inside of the sleeve, and everything goes inside of the tubes. Here you can see the rack. And you have this for 50 ml tubes and also for 100 ml samples tubes. Here is just showing the layout of the protocols that you can set up, and it is going to calculate how much of buffer depending on your protocol you need. And just I have just one or two case studies here that is showing the comparison with beads versus resin. So this is for protein A magnet beads, and using the same equivalent of the resin. So as you can see here in this slide, we got a little bit higher yield for the resin in terms of the magnet beads used. And you can see here the purity of your purification. Sorry. Yes, I think that is I don't know what's okay. Now that's the end of my presentation. I just wanted to also tell you that we have a lot of other instruments, not only the AmMag Quatro series. Also have instruments to stain your maxi-stain your samples. You have the e-blot for transferring. We have gels. And you also have the cytosync for cell purification. Yes, that's the end of my presentation. Thank you very much. And you can, if you want to, you can visit us. We are at 404 booth.
Thank you so much. Are there any questions from the audience? Okay, I'm coming.
Hey, great talk. I just wanted to know if any of these machines, any of this automation is limited by the starting culture. Like, is there any upper limit or lower limit on the ODE or in terms of pellet weight, anything?
Yes, exactly. Good question. So if we're talking about the maxi prep instrument, the maximum amount of cells that you can input is 1.5 grams of pellet. More than that, we are not going to get any more plasmid. Actually, you are not going to get plasmid because of the physical limits of the instrument. So we would need to use more of the resuspension lysis buffer, and that is not possible because everything goes inside of a 50 ml tube.
Awesome. In the interest of time, we're going to move on. So can we please thank our speaker one more time?